Sure. Awesome. Hi, everybody. It is the 29th of September, 2021. This is our second GBM for the fall term, Drexel Society of Artificial Intelligence, Drexel AI. And we're going to continue talking about our project, our proposed project this year, or this term, which will be in radiogenomics. I'm particularly interested in doing this project, considering that I'm working on a grant that is kind of in this field, in a related field. We're working on merging together, imaging data, genomic data, epigenetic data, physiological outcomes. We're trying to take the big, which is what you can observe, what clinical data you can observe from a patient, and connect it to the small phenotypes that a doctor otherwise would not have access to unless they profile the blood of the patient. Fun fact, I can't give blood. I can, but I pass out. What'll happen is they'll take my blood, they'll be finished taking blood, and then I'll just pass out after. So it's after the action of blood has been, after like the action of taking blood has been taken, I faint. So do not ask me to give any blood, because I will not do it. Yeah, so for everyone who doesn't know, my name is Ethan Moyer. I'm the president of Drexel AI, and I'm a third-year biomedical engineering student. All of these videos are uploaded to YouTube, so you can feel free to just check them out afterwards. The way that Drexel AI usually goes is that we do a project a term, or a project or two a term, focusing on a topic. We've done topics in neural network optimization, time series prediction, the stock market, protein structure prediction. This term we're doing medical imaging and genomics. That's why I called it radiogenomics earlier. Before, I know Safik has something prepared. Before he goes into that, I want to just mention a couple of things. So I said we upload all these videos, and these videos are all on my YouTube channel. If you want the link, you can just ask me. I used to be really good about posting the link to the general chat afterwards, but I haven't done that. So last week, to recap, we found this data set, thanks to Sepek, that is actually particularly new. I mean, June 1st this year. And I downloaded all the data, and it's about 100 gigabytes. So I have it on an external drive. So what takes up a lot of the data is the segmented DICOM images, which I think are the CT scans and the PET images as well.
PET. Yeah,
PET CT. There are AIM annotations. I don't know what AIM stands for. Probably clinical annotations regarding the patient. Clinical data, that's a smaller file, so it probably isn't as robust. What I'm imagining the clinical data is, Savek, is just like 1 hertz data.
Okay.
My company deals with higher frequency physiological data. Like we deal with the ICP waveform. For people who don't know, ICP stands for intracranial pressure. It's the pressure that's pushing up against your cranium from underneath your skull. And you can sample that at 1 hertz or once every second, once in every minute. But when you actually look at the waveform, it behaves in a really periodic way. This 3-peaked waveform tends to repeat, and you can actually.
I'm sorry to interrupt, but you mean the skull on your pressure? I mean the pressure on your skull isn't constant?
No, it's not. It changes.
Damn.
Yeah, it's a function. It's a function of the oxygen in your blood. It's a function of the compliance of your brain. And clinicians use the relative sizes of these peaks to determine, brain compliance. I think if like the second peak, P2, is higher than the first peak, then that means your brain is more compliant, which is bad. I think that's the interpretation. So that's the kind of physiological data we're working with. And obviously, if you're dealing with ECG and a sample that, once a minute or once a second, you're not going to get the waveform that you'd see in the EKG. EKG is, you have the QRS wave, the S wave, the QRS wave, the T wave. Look at ECG, EKG, whatever. Anyway, we want to have access to this data. Yeah, the QRS, the P and the T and the U wave. But I'd imagine that this data is just, pretty low frequency. And then there's RNA sequencing data. For those who don't know, the gene expression omnibus geo is what controls a majority of data sets related to genomics. And I use this for methylation data through my lab. It's hosted through NCBI. So we found this data set, and Savek was also good at posting 2 reviews, which I read, that kind of talked about, radiogenomics for precision medicine and what has been done so far in the field. And after a while of doing this kind of stuff, reading papers and, learning about new topics, you can kind of tell when fields are at their infancy, because they talk a lot about mechanisms that really haven't been validated with data. And just from reading through these papers, I can tell that there's a lot that hasn't been done. For instance, this review, a majority of their results for the review was just a summary of different imaging modalities and the architecture that they use. And then they looked at, I think it was either this one. Yeah, they looked at, what kind of architecture was used for what percentage of studies. For instance, everyone's using, oh, sorry, this is not what the one I wanted to look at. This one, yeah. Everyone's using a convolution neural network here, right? There's this thing called a capsule net, which is actually particularly interesting. Basically, instead of having like input features into a neural network, for those who don't know what a neural network is, it's basically it's a graph, and each node in the graph is a feature, and then the connections between nodes are weights, and these nodes are augmented in layers such that the data starts at one side, the left side, as inputs, and it kind of propagates towards the right side through some equation that's like a weighted sum between each input and the weights from the line. So you'd have a weight W for each of these connections into the second node in the second layer. And then using an algorithm called gradient descent, you can actually back propagate and adjust the weights based on your error for the final outcome. And we can go more into the theory behind neural networks, because I love talking about them. But yeah, so a CNN uses the convolution operation. The convolution operation is a way to compare patterns. You can kind of think of it like that. When 2 patterns are matching, the convolution operation is at its maximum. The capsule net actually doesn't work in terms of neurons. It works in terms of what they call capsules. And these capsules are like predefined objects that they recognize. So in the article I read, they were talking about like, a house that is composed of a triangle and a rectangle, but then also a boat, which is kind of funny, comical. They also made it in terms of a triangle and a rectangle. The difference between these two is not the shapes or the features, it's the orientation of those features. So with neural networks, you tend to lose because of this operation called max pooling. It's basically a way to adjust the dimensionality as you go from like the input space to the output space through different layers. Because of max pooling or any pooling techniques, it's like a weighted sum or non-weighted sum. You tend to lose the orientation information as you propagate from layer to layer. But when you're dealing with capsules, you're moving the relative the features from each layer, but then you're also keeping track of the position of the objects, which I thought I've never heard of capsule net before, so I thought that was particularly interesting. Then there are some off-the-shelf models that people use. That's the pre-trained R. RNN is a recurrent neural network that's used for time series prediction. It has some sort of memory. It remembers the previous state. It's like an artificial neural network, but it has memory associated with it, so it can remember the last state it was in. And an autoencoder is really cool. It's a way to an autoencoder is composed of 2 things: an encoder and a decoder. And what you usually do is you usually take an input and you take an image, you could feed it through the autoencoder, and you try to encode it to a smaller representation of that image, and then you want to decode it into a reconstructed version of that input. So then what you have is you have these two models. The one on the left acts as a way to efficiently encode data, and then the one on the right is a way to effectively decode data. So, all things aside, this is what most people in the field are using for like algorithms to look at this type of data. The second review talked a lot about cancer biology. I also want to, before I go into the whole cancer thing, their figures were comically very similar. They looked almost
I've actually
identical.
described in my notes, I've described the second paper from Frontier, so that would make it easier
Okay.
to
Yeah.
explain.
Yeah. So what people talk about for radiogenomics is they have this acquisition of data. The top part is the genomics. This is a chord diagram. It's to represent like set overlap. So this is probably relationships between different samples. The bottom are different images like CT or the PET scan. The second part is selection. So this is kind of like creating a feature set which properly describes properly describes your input space. Like you really don't want to throw an entire image, like a PET or CT image, into a neural network, because those images are pretty high resolution. So it's pretty computationally expensive to like just analyze all of those images. The second one is association. So they're trying to look at the association between outputs. So can images predict a given phenotype observed on the genomic scale? Then analysis, and then the endpoints are is taking what you've analyzed and constructing biological pathways and doing this grading. And I think this grading is kind of like, you can kind of think of it like assigning cancer stages. Like we know that stage 4 cancer is a lot worse than stage 1 cancer, and people can put individuals into different cancer groups. Where that isn't evident is in a field that I study, relating to traumatic brain injury. We don't have that grading. We can't assign a patient. We can't assign a patient in more granular groups besides severe, moderate, or I guess mild traumatic brain injury. A person in a coma. We throw around the word coma, but there are definitely different coma states, and there's definitely people have explored like degrees of consciousness, or are they called degrees of consciousness? DOC. I forget what it stands for. But yeah, we'd like to be able to do that grading in other settings besides just cancer. So yeah, that's kind of what we looked at last week, and we have a data set. That's probably the hardest part. We have what people have done in the past, right? A lot of them have done convolution neural networks, and I kind of want to do something different. Depending on the data we have. I might want to look into sparse coding again, and I'll explain what sparse coding is in a bit. But me and Alicia, we have experience with sparse coding. Data set and a data set and a perspective method and a bunch of eager Drexel AI members to tackle the problem. And usually what happens is we do research, and then we write a paper, we get results, and then everybody who is contributing to the paper is put on as an author, and then we try to publish that to either arXiv, which is a unpeer reviewed database hosted by Cornell, for which you can go on Google Scholar and you can search up your article. It's kind of cool, get your name out there, and it will submit those to conferences and such. And we submitted maybe three of our papers last year to a conference. Sorry, to 3 different conferences, which is pretty cool. So, anybody have any questions about the topic, or?
Yeah, I have a question. Like, what's our main goal with this like data set? Are we predicting or classifying something? Are we like grouping things? Or are there multiple?
Okay, so
Sure.
should I go?
Yeah, you
Can I
can go.
So this whole field is called the precision medicine, and it combines three outcomes. So we are looking towards predicting the diagnosis and detection, combined with how the disease is going to prognose over the period of time, and the treatment decision. So these three are the main objectives of the project.
So the first and the third are predictions, and the second one is more like series modeling of how it'll like affect the body over time.
Yeah.
Okay.
We could really choose to
Yeah.
do any one of those three. I'd hate to tackle all three at once. I think that'd be
Actually,
difficult.
I was looking at different models, and you could perhaps do 2 models and get all 3 done.
Okay.
Yeah, I'll
Well,
send you a few papers.
yeah. Do you want to go into your presentation? I mean, Alex,
The
that's definitely a good
notes.
question, and that's where we're going next, right? We have to decide what exactly we're doing with the data set.
Yeah.
I have it downloaded on my computer. I haven't like loaded into Python. I'd imagine that I'm gonna have to do some sort of batch magic with the images.
Yeah. Also, how many of you have never like done any work with machine learning or have no clue what machine learning is? Like, you could raise your hand.
Abigail,
Abigail,
Walter.
you could raise your hand. You could raise your hand through Zoom. Okay.
Good.
Okay. I'll give you a brief introduction.
Yeah, thank you, Savek.
Okay.
You're better at that than I am.
Okay, so I'm sorry, you guys, I wasn't able to make a presentation. I just have notes that I made during classes. So forgive me for that. So I have just done a review of a review and just added a few more points from here and there. I'll be jumping between Chrome and these notes. So basically precision medicine. So it follows like a one size fits one. So basically each person has a particular way of treatment which would benefit that only individual. So it helps in the diagnosis and prognosis of that person. And it has been seen, like especially at Penn, there is a whole center for precision medicine, and they have seen how the medical outcomes have gone, really gone up, and how the mortality rate is going down. So these therapeutics are planned according to the genotype and phenotype data of that particular individual. So to retrieve the genomic information, there are a few ways to do that. I'm just covering a bit of the biological part because we need to know the background before the project. So we sometimes take the biopsy of the focal tissue, and this is like CT guided. So you do a CT and you know where the tumor is, and then basically you put like a fine needle in it and take a pertinent part of the tissue and get the biopsy done. That is the one way. Then this is a different way of microarray genetic analysis, which is also present in our data set. I really don't know how to explain this because this is not my field of study, but Ethan might know what this is. Do you know this?
Is that RNA-seq or?
Yeah, I guess.
I'm familiar. I'm more familiar with microarrays in terms of DNA methylation, not so much gene expression.
I guess
but
it goes the same way. Like if it is expressing its green, if it's not expressing its no, if it's expressing it's red, and if it is not expressing it's green. Yeah. So yeah, then we go into like what the whole model looks like. So you first of all, you take an image, you get the image acquisition, then you do an ROI segmentation, which is region of interest. What areas do you specifically need? We will talk about these. And then there are more radiogenomics features such as shape, histogram analysis, texture. So all of these features are available in the data set. And then we make a statistical model with all these features and patterns and get treatment outcome. So we start with the acquisition of raw data, which we already have. We are working with the PET, or we call it the positron emission tomography, computer tomography. We're working with that, and it basically, the common uses are, first of all, detection or make diagnosis, then determine if the cancer is metastatic. This is a really good point because this is really important for us while making predictions. Then assess effectiveness of treatment, which involves the prognosis, and determine if the cancer has returned after treatment. So we'll see that some patients have a follow-up after like 5 years or 6 years in the data set. And then we could also use that data to see if the cancer has come back after a surgery or something. Right. So this
Can
is
I ask a question? So is the purpose of this to return like a statistical value for like how likely it is that the person has cancer and like information about
yes.
it and
It-
how
it-
bad it is, like the
It
severity?
can be. It can be.
It can be.
We haven't decided what what the outcome measure is going to be yet, but that's one option.
That could be one of the options. Yeah. So this is what a PET CT scan looks like. You could see it has the cancer, and one of the very good, like this one was a really nice one. This is a CT scan, and this is a PET CT scan. So PET CT scans look more like a heat map if you have worked with one. So it precisely gives you where exactly the cancer is, so that the ROI segmentation is much more precise with it. And then there's a pre-processing, which is all done in the data set already, I saw, because they gave a segmentation, the AIM. So yes, so this is called the region of interest, the ROI segmentation. It helps us in getting a particular point in the whole image, which so that we could get homogeneous and reliable traits. And we are not taking account of a huge space and getting biased results, because also considering the ROI, if it is too small, we may not get enough information about the voxels for analysis. And if the ROI is very big, the work we are doing may get biased because the heterogeneity of the tumor, right? After that, so the segmentations we have, it is, I actually emailed them to ask this, and there are 3 types of segmentations. One is manual, in which a clinician would sit and do the segmentations by hand. It is time consuming and not very reliable because different clinicians do it different way. Then there's automatic, which is fast, precise, and cheap. And the state of the art, whenever we say state of the art, it means like one which is currently being used and is like one of the most advanced methods out there, which is the semi-automatic segmentations, where we keep a clinician in the loop. The automatic segmentation is done and the clinician sees whether the segmentation is done correctly or not. And one of the most used models for that is the multi-parametric Gaussian mixture models, which are easily available online and could be used.
Is that the same thing as is like a mixed model, or?
Yeah, it's a mixed model. You could look that up. This is what was really interesting for me and would help us. So these are the features, a map of features that we get out of the whole radiomics. So there are, I would send this on the channel as well, but there's static and dynamic. The dynamic is mostly, you could get this from your CT scans and your PET scans, and then there's static data, the morphological data like volume, the surface area, how sphericity of that, then their intensity, and there are like a bunch of different features which we could use. And apparently we do not use all of them because using all the data make it a bit redundant. So we just use a set of, oh my God, okay, yeah. Just a set of features that we feel would be interesting to use. And we actually, now that we are hope to work with UPenn, we could get a point of view of the clinicians that which particular features are important in making decisions, which would help us analyze more. So yeah, and for those who don't know what deep learning is or machine learning is, so it's basically a subset of, deep learning is a subset of machine learning, where we use a technique called neural networks with multiple layers to construct an architecture based upon mathematics to form correlations and detect patterns in a training data. So this is a very basic representation. So in a traditional computing, you put in a data to the computer, and you put in a program to the computer and it would give you an output, right? But in machine learning, we say we give the computer data, we give them an output, and it gives us a program. And then we use this program on further data to get an output. So our main goal is always to find a program rather than finding an output. If we look a bit mathematically, imagine we have a data set which has these multiple points in xi, yi, where xi is the input and yi is the output. And then there is a different distribution, which I like to call the Gauss distribution, because nobody knows what this is. And this is completely different and has the model has never seen it. So it is xn and yn. So in the training phase, we insert these xi values and xn values, xi values and yi values to the machine learning algorithm and get a function h. And this function h is performing good if it is if h of xi is close to yi, right? And then during the test, we put in this Gauss, pull up a point from Gauss distribution, put it into the same function and hope to get a yn. So this we are making to make a generalized equation. So the main objective, so the lung cancer has a really high mortality rate and accounts for like 13% of all diagnosed cancer. Lung cancers are generally of two types, non-small cell cancers and small cell cancers. 85% of patients are with the small cell, the NSCLC, and we are working with this one. And apparently this has a slower growth rate than the small cell lung cancer. And it has like a distinct disease with genetic and cellular heterogeneity due to different mutations. That's why radiogenomics is generally used with non-small cell lung cancer rather than the small lung cancers. And then biopsy alone cannot give complete analysis of this cancer. So that is a point to keep in mind while making the decisions. And yeah, we would be there for utilizing deep learning algorithms to combine various features and give predictions about one of these three, or maybe two or three outcomes, which is diagnosis, detection, prognosis, and the treatment decisions.
Do we want to look at the data? Well,
Yeah.
first off, does anybody have
yes.
any questions?
Wait, I have data opened. Wait.
You downloaded the data set?
No, I just opened it online.
Oh, you can open it on.
Can you see the screen?
Yeah.
Okay, I think it's working. Okay, so
Is that a DICOM viewer or something?
Wait, how do you do
Yeah.
that?
Oh, it's in the cancer imaging archive. You just
Oh, okay.
you just click on this,
Oh,
and yeah, and that's
is that
a
what we
viewer.
downloaded?
No, you don't have to download it. It's online. So you
Sorry,
could
is that the data set we're working with? Is what I'm asking.
Yeah, that's the same data set.
Okay, I see.
Yeah.
I was just looking on a computer, so I'm trying to understand it myself.
Yeah, it's the same data set. So this is one of the sample scans. And you could see
The human.
the human, yes. Apparently these are the lungs, I guess.
Yeah.
Here. And you might see, so here, if you zoom in, if you try to zoom in here, you will see, oh, how do you zoom in? Here. You will see these white matters. These are
Yeah.
the cancerous cells right here.
Where? Can you go back to that? I want to circle that.
What? Oh, wait a second. I'm trying to. This is really hard though. It's hard to control.
Right here. Right here.
Oh, right here. Yeah.
Okay.
Exactly.
Cool.
Right.
Yeah. So
There might be more. This is what I could see of not being. See this one, the flare here.
How do you
Okay.
know?
I studied.
you did work with this before.
Then
No, I mean, how do you identify the cancerous cells from like the general lung, gunk, whatever?
so we're not actually looking at cells, we are looking at the tissue right now. So wait a second, let me pan in. So do you look at this here, this white kind of flare-like looking thing?
Oh, I see.
It shows the abnormality in the tissue, because the normal tissues would look like something like this, generally black in matter, but these white ones are saying that they have a fluid in it, which is generally called the edema, and the middle part is called the necrosis. And I'll probably send some stuff on it on the Slack channel so that you could get a very brief introduction to that. And yeah, so this is the data set, if anybody doesn't know about this. This is the data we are working with. We have 211 subjects. We
I
are working with.
downloaded it, so I was trying to figure out like how we're gonna actually use it, like the processing, and I guess I'll have to figure out the details.
Yeah. And these are the two labs we are currently talking with. It is the Artificial Intelligence Biomedical Imaging Lab. It is under the Center for Biomedical Imaging, Image Computing and Analytics, CIBICA. And we are trying to work with them. There is Professor Ditovich, Davidovich, I don't know. I call him Ditovich, I don't know why, or Professor Christos. He's really interested in working with us, and Dr. Young, he specifically works with lung cancers. So he would be helping us throughout these projects. We would be hosting a meeting soon to discuss a bit with them about how we should go with this project.
Yeah.
And then we have this second lab, which is the computational biomarker imaging group, which could help us with the annotations and the ROIs segmentation, which exact places to look up for, what exact features to use. So I think that would be really beneficial for us.
Would be. Thank you for putting that together for us.
Yeah.
So yeah, cool. Does anybody have any questions for Sefek? No. Okay.
I had one question.
Sure.
Yeah.
For it.
So is it all just lung cancer, or is it all types of cancer that we'll be looking at for this study?
Just lung cancers.
Yeah.
Okay, cool.
And we have associated metadata for whether they were smokers or non-smokers, which is like great.
We have that?
Yeah, we do. I was looking, there's a metadata file, so that's why I was looking in the data.
That's nice.
Yeah, it is nice.
Don't smoke, guys.
Yeah, it's not good.
Yeah, it's not good.
I want to show the stuff. Just gotta make sure I have it all open. Shifted to, what do I, two versions of these? Oh, sorry. Okay, so where's Zoom? I lost Zoom. Here we go. Okay. Cool. So yeah, that's a good introduction. I just opened it up in Python. This is an example of the CT image. We're not viewing in like a dynamic viewer. I just have it open with matplotlib. There is this, I mean, there's fancy software out there, but to view these DICOM images, but you can use something called PyDICOM.
Yeah.
And if you Google DICOM images in Python, it's pretty easy to just like load one in. This is an example of a cross section that Savek was demoing. And this is for the patient 1-1. And if you look, so I downloaded all the data in a external drive of mine. It's about 100 gigabytes big, and we're going to look into getting a Google Colab space with 2 terabytes of storage space for the club. I haven't done that yet, but we're going to be doing that. Anyway, so yeah, AMC 001, that's a single patient. So when you go into the metadata associated with that, you can see that, you know, each patient has an associated metadata, I think. Sorry, I'm trying to find the key column. Yeah, right here, AMC1. And you can see that what study they were in and when the study was conducted, number of images, file size. This isn't at this metadata isn't at the patient scale, that's at the image scale. When you go to here, you see AMC1, you see it was a male, it's 34, wasn't a smoker. They had some other information like whether the tumor was the tumor location was checked, the histology. They even have some mutation statuses, so they're looking specifically at these genes. Data recurrence for the tumor, when he died, days between surgery. You can tell there's a lot of features that we could use. It's pretty sparse because not every patient has the same data, but that's the kind of data we're working with. So yeah, this sample sheet is what controls it for each patient. So these AMC numbers, and then the metadata here is what controls the images taken for a given patient. So this patient had, what was it, like 6 sets of images, and then for each set there were a different number of images. I think like this one is 150, this one only has 1. I think this one was the entire body. That's the one I looked at first. It wasn't cool. So this one's, in my opinion, much cooler. In addition to the images, the metadata about the patient, the metadata about the images, we also have data about the gene expression. So I mentioned that, I mentioned that, you know, when you go to the website, there is RNA sequencing data. RNA sequencing is a way to measure the relative frequency of different transcripts that are expressed from a cell. I wonder if there's a way that I could show this. Yeah, so you sequence the RNA in a cell, and you can align that to the genome and then get a sense of how many transcripts there are of a given gene. And then you can assign, okay, you know, if there's a bunch of transcripts in, you know, right here, like if this is the maximum, if this is a peak, then this gene is probably being highly expressed. If not many are aligned right here, this gene probably is probably not being very much expressed. And we can get that data across different patients. And the data kind of looks like this. Each row is a gene. Each column is a patient number. I think these R01s correspond to different patients. There's probably some mapping between this ID and the AMC ID I presented earlier. I think if I do, sorry, is that gonna work? Yeah, it's right here. So this now it looks more like a matrix. You can see that each one on the left is a transcript or a gene. And what we can actually do is we can take one of these, go to NCBI, under their gene search, and you can look at what these genes are. Yeah, homolog 4 lysine demethylase. This is demethylase is, you know, unmethylating, is an unmethylation enzyme. And there's a lot of information. You can see it's not really tissue specific. It's expressed in a variety of tissues. I don't know where the site was taken. The site, like where in the body the RNA-seq was. Oh, sorry. No, I don't know where in the body the RNA-seq was conducted. Okay, so the RNA-seq was extracted from the tissue where they may have thought that the non-small cell lung cancer was located. Yeah, so we can look at like, you know, LC-W. Oop. Sorry. There are about 22,000 genes in this data set. I don't know how many samples there are, however many columns there are on here. Yeah, but this is the kind of data we're working with, and it's pretty. This has to be like slightly pre-processed, but you can basically use this outright because it's a matrix. The imaging data is, and you know, the metadata is pretty simple to use because these can be treated as factors. I mean, smoker versus non-smoker is binary. Ethnicity, if it was available, you know, you could use that as well. Male and female, age, you can use that. But the most difficult part is the image segmentation, in my opinion. They said they were segmented images, but they just lied about that. Because at least the ones I looked at aren't segmented. So Vec, did you see any segmented images when you were looking on it online?
No, there wasn't any, but they
Yes.
gave you the segmentation, right? So you need to implement that.
Where did they give that to us?
They gave it to you, the AIM segmentation.
Where?
Oh, wait, when you look up on the data set, you'll find some images have the segmentation done. Wait, let me show
Only
you.
some of them? Because I'm looking, do you think, oh, wait. I think that's controlled in this metadata sheet.
Can I share my screen?
Yeah, I'll do some looking while you're doing that. Stop share.
Mhm. So when you look at the summary of the data set, so we have CD, we have PT,
Yeah.
and we have the segmentations. So we have 144 segmentations.
So
And
not all of them are segmented?
I don't think so.
That's silly.
That is.
We could do our own segmentation just so all the data is segmented.
Yeah, we could just get a mixture model, put in the data set. We could actually train our model on the segmented data and do the segmentation, the rest of the segmentation at the same time.
Oh, that's a good idea.
Like this person.
Yeah.
Okay.
No, that's a good idea.
This is a segmentation result.
So just not to brush over what Savek said, he said that since a third of the data is you know, segmented, what we can do is we can train a model on that segmented data and then apply that model to the other ones. What do we mean by segmentation?
Can you open this file, Ethan?
I was going to open this one, show her this.
The
So
segmentation one.
This one, the resolution of this image, I don't know what the resolution of this image is. It's relatively big, I think. So yeah, you can imagine that not all of, okay, so in machine learning, we deal with features, right? And we want to incorporate only the most important features. So in a CT scan, right, this is the image, and you know, what are some features you don't need? You don't need everything outside of the circle, right? That's the easiest one to spot because that doesn't change through from picture to picture. If we're trying to predict whether someone has lung cancer, if that's a basic idea, then those features don't tell us anything about whether they have lung cancer. That's just the artifact of it being a CT scan. In other images, you might actually get the full square, but in this case, we're not going to get anything in the corners. You know, other things you might want to get rid of is anything outside of the body that doesn't really have much to do with whether or not they have lung cancer, because lung cancer often operates inside the body. Often it happens inside the body. So those are very obvious things that you would segment out. You would just remove those features. So when we say segmentation, we just say that means like to just focus on the features which are very important. Alex asked the questions, and I can't move. What did Alex ask?
Can you share the data set link? He found multiple versions of it, multiple older versions of it online.
I'll send it.
Yeah, we'll likely use the newest one because that one doesn't have many that has the least number of publications associated with it, if any. So the question becomes, okay, so if we're focusing on, just this part of the image, what is what of these features, what are the best that would help us determine whether or not someone is going to have lung cancer? And that could even be our outcome. So that, I mean, that's pretty simple.
That's pretty simple.
I know.
Yeah.
I think, but to be fair, if we can implement that, then, if we can implement the pipeline to do that prediction, then, just changing out our predictive variable isn't that difficult. So.
Well, that's facts.
Sorry?
Yeah, we could do that.
Yeah.
Yeah.
So, you might segment this further and you might say, oh, anything that's inside the lung we should take into account, right? And that might be different from image to image. I don't think it could be different from image to image because you need to have the same feature space from image to image, like sample to sample. So you'd want to segment out just the lung from each image. And these would be the regions of interest. And then you only use this segmented image to predict whether or not someone has lung cancer. Like, you don't need everything else. You don't need anything outside of these red circles. And these would be the segmented images. So that's what I mean by segmentation. It's used to reduce the feature space. Yeah. Cool, good question. So yeah, this is what the data looks like. We can really do whatever we want with it. I mentioned, predicting using this data, right? The images, the CT scans, the PET scans, and the metadata, so whether or not they're a non-smoker, to predict whether or not they have lung cancer. I don't know if that is anywhere here. It probably is.
But that won't include the radiogenomics part of the data set.
It can.
I mean, that won't be much novel.
Do you have a better prediction, better idea then for what we could do?
I mean,
didn't you mention like you want to do like survival prediction?
Yeah, the prognosis.
Yeah.
That could be fun.
Okay.
Is the outcome of this project supposed to be an AI that can like predict things from cancer scans, or is the outcome supposed to be to use an AI to like write a paper, a statistical study or something? Sorry, I worded that really poorly.
The outcome.
I meant to ask, is the outcome going to be the AI itself, or is the outcome supposed to be using the AI to make a prediction about something, or are you undecided on that?
So right now, the AI technology, we haven't decided what we're going to use. That's more or less up to us, like by design. Depending on the problem you pick to tackle, there's different methods that are better suited. Like if I was trying to predict, like just by nature of the problem, the first issue is finding the data set. The second issue is determining what you want to use that data set for, and then the third issue is determining what machine learning algorithm you want to use on that data for that problem, because that's dependent on the first two. The result of this won't necessarily I mean, if we do research and it works very well, then maybe the outcome of this will be AI that is really good, and we sell it and we become millionaires. But the likelihood of that happening is little to none. So the outcome of this will likely be just a exploratory effort to learn more about radiogenomics to help you guys out, to help you guys and teach you AI. But Savek and I and Alicia, and I'm sure other people have pretty extensive experience with AI. So we're hoping to teach you guys how the ball gets rolling around this place with machine learning, if that makes sense.
Yeah, thank you.
Sure. So Vivek, what does the survival analysis look like? Do you wanna share something? I always see it. I mostly see it with mice.
I need to pull it up 1 second.
Is it just based on factors? It's how long are they gonna live?
Sometimes.
Okay.
Like it depends on the model we use. So sometimes
Okay.
people use they would first use a CNN,
Yeah.
a deep CNN. They would get the imaging part done from that. They would get a result, and then they would use that result combined with the other genetic informations and make a graph kernel. And then the graph kernel would predict the survival percentage, whether the treatment is going along right or not.
So when you say survival percentages, can that be related to like what feature is that related to? Is that related to like the time of reoccurrence of a disease or time till death or?
Yeah, the clinical data.
Okay.
Yeah. Wait, I think I shouldn't show you guys this, but okay, who cares? Nobody speaks about this. So this is one of the projects I did at Georgetown University. 1 second.
Yeah.
We did something very similar. I'm trying to find it.
I am recording, so just letting you know.
Oh shit.
Yeah. Okay. Just
When we stop
I don't
when
want
we
to have
stop recording.
to. I don't want to have to edit the recording because that's annoying. I saved him.
So I'm not
Yeah.
showing.
Yeah, that's fine.
Yes.
So like maybe I can draw something. I love drawing.
Right.
Survival
I'll
analysis.
pull up a paper.
Yeah. Survival analysis, you can imagine that you have this curve. And I think the idea is that you have like this, I think you have these two axes. And is this right, Savek? You have something like this, these curves. What's the x-axis?
It's very different. You get a percentage. It's really interesting.
I'm looking up an image.
Try searching graph kernels.
Survival analysis first. As you can tell, there's stuff that we don't understand completely. Yeah, so this one. Yeah, so over time, the probability that the given person like will survive. Obviously, you want the survival probability to be as high as possible, right? You want it to be up here, and you can determine the probability of them surviving based on the clinical data. I don't know if you could do it through time like this because we don't have our data isn't distributed through time. That's a problem.
I haven't looked up at the clinical data.
I don't think it's.
I'll do that today.
But the images aren't. The images aren't.
Or a period of time.
No, it's probably just one image at one point. So you can't do a survival analysis like that.
Yeah, like we need a time analysis for that.
Yeah.
Wait, let me see how the clinical data looks like. 1 second.
What was messaging me?
Oh, yeah, I found an article. It's called "Long-Term Cancer Survival Predictions Using
Cool.
Multimodal Deep Learning."
Would you need images through time to do that, or how does that work?
I think you would need over a period of time.
Alicia,
That's how
no,
you see the growth.
Okay.
No, they have given no time.
It says notable examples include methods designed to use clinical and gene expression data. Other methods focus on imaging data.
Yeah, but we don't have that through time. Or maybe the curves are learned. I don't know. That's kind of tricky. I don't understand it as well as I'd like to.
I have one more idea. We could do a detection using a particular genomic feature, like detection of lung cancer using say any sort of this KRAS mutation status or something like that.
Okay.
Like incorporating new features into the detection process.
I have a step further.
I mean, this data set is so good. They have chemotherapy, radiations. This is actually what I wanted to do with that. Once you get all the features in, it predicts whether the person needs a chemo it does not need a chemo, or it needs both? That's what I wanted to do.
Is there any way that we could, okay, here we go. Is there any way that we could impose a grading on the images? Say one patient has a more severe cancer, and we rate their cancer severity on a scale from like 0 to 1, or like we could bin them into different, like 5 different bins based on like no cancer, low risk, medium, low to medium risk, medium to high risk, or something like that, and then classify that based off of the genomic data.
I'm not sure about that. Have they given the stages? What's the stage of each
I don't
patient?
know. I don't think they did.
I'm looking through the clinical data.
If you
What
if you
they
look
have-
in it's in the data labels.
So what I see is like they have given the treatment, they have given the chemo, radiation, recurrence,
Recurrence.
yeah, and then
I don't
date of
see-
recurrence.
I don't see.
Date of,
You could predict recurrence, time till recurrence.
Oh,
yeah, date of,
Abigail wrote you could record probability of relapse based on severity if you could do that.
Yeah, that is a
that's
good idea.
good one.
That is a really good
That's
one. I like that,
that's a good
actually.
one.
I think we should do that
We could do that.
because we have relapse, right? We have relapse data, and we could use the genomic data as well as the image data.
Yeah,
We wouldn't
To see
need to focus on giving we wouldn't need to focus on providing labels, because the labels is the relapse. It's yes, there was a relapse, or no, there was a relapse.
no, there wasn't a
Um,
relapse, right?
So would it just be a yes or no, or how would we deal with the probability if, would the probability be high if the relapse was closer to the given date when the
One.
image was taken, or would it be less because the relapse is farther in the future? Like, how do you treat relapse? Are you going to treat all, like, any relapse as binary as yes?
Yes, that's how it
Okay.
works.
Okay. But then there's
is
no,
called-
you're going to
Well,
relapse.
that might vary a little bit based on how they were treated too, right? So the data doesn't really account for that, right?
Yeah.
Like how effective the
Yeah.
treatment was.
It is
I don't know.
because this disease is not metastatic. And because this is relapsing, it becomes metastatic after the surgery. This is really interesting with this particular non-small cell lung cancers.
Can you say that again?
So basically the small lung cancers, they tend to be metastatic. That means they tend to grow out. But the non-small cells lung cancer do not show the property of metastatic conditions. But here we see that some people did have reoccurrence of cancer, which is really interesting to see. So if we could predict the recurrence, even though the disease is not metastatic, it is really helpful for clinicians.
Is that a function of like the images and the genomics, or of their intervention? I think that's what was brought up. Do you know? I mean,
What?
You see, the variance associated with recurrence can that be attributed to the genomic and imaging data we have, or is that attributed to stuff that happened during surgery?
Oh, we could just add it to the genomics and imaging. We could just add it to that.
Yeah, but then we're assuming that every intervention, if it was chemo, every chemo was the same, which might not be a good assumption. Oh, they had chemo.
I just have a general question I'd like to ask. I gotta go soon,
Yeah.
but the data set you send has like about 200 or so patients. Are we supposed to, like, for our two predictions and our time series like prediction as well, are we supposed to generalize to those patients in that hospital specifically, or is it just like patients in general? Because we don't really know like how the data was like sampled at all.
Yeah, that's a good point. There's definitely gonna be bias that's associated with this. There are different models that are robust against overfitting. That's kind of what you're worried about. You're worried about overfitting for a hospital. and there's models that you can use. I know random forest is pretty robust to overfitting because it's a consensus-based model.
SVMs are not.
They're not, they're bad. Yeah, I know everyone's leaving. Bye Doom, bye Brandon, bye Bennett. Dropping like flies. Thanks for coming, guys. Cool.
Thanks for coming, guys.
Yeah, I'm really
Actually-
excited about this project.
Me too.
Alicia, sorry, not Alicia. Abigail, I like your idea and I'd like to do that. But Savek is kind of taking the lead of this project, so it's up to him.
I'm hoping the UPenn professors give us like good guidance
Exactly.
on how to go
That's
about
what
it.
That's what I'm actually waiting for, because these guys have been doing this stuff like since 2010.
Especially
They-
the one that like is good at lung cancer, like specializing in the lung cancer imaging.
Yeah, Dr. John.
Yeah.
Yeah.
So,
Thank you guys for hosting this.
Yeah, we'll see you
Seems
next
like a fun
Wednesday.
project.
thanks for
yeah.
coming.
Yeah,
Thanks
thanks
for
for being
coming.
invited. Cool.
Ethan, also about the involvement fair.
You're gonna be there,
I'll-
right?
Yes, but I'll
Okay,
be like,
cool.
I'll be coming at 4:50. I have a class.
That's fine. I have to leave at like 5:30. I printed out
Oh.
stuff.
You did? What did you print?
Yeah. Oh, Adine. Oh, Adine, are you there?
Hey,
Hey,
how are you guys?
been quiet. I didn't know you were here.
Yeah, sorry, I was lurking.
Oh, that's cool. You're
That's
gonna be
cool.
involved in the project? You want to be?
Honestly, the project doesn't seem too exciting to me. And
That's cool.
well, my senior project is very, very similar. Did you
Sure.
talk to Kevin Carnani by any chance?
I think
He was
in
here
our
last
meeting.
week.
Yeah.
Yeah. Also, actually, what's it called? Do you know Riddick?
Yeah. We
Yeah.
said Moberg.
Yeah, exactly. Yeah, he's in my senior project group. And we're doing
Oh,
something pretty
cool.
similar to this.
What cancer are you looking at?
Sinus cancer, I believe.
Okay.
Oh, that's
Are you
nice.
using CT or using genomic data too, or is it just imaging?
Just imaging.
Okay.
Mm-hmm. That's
Yeah.
cool.
Yeah. Good
What-
luck.
Thanks so much.
Over
Talk
this
to you guys
week,
later.
I'll try to look into more data, talk
We
to
should-
these professors
wait.
more.
Hold on. We should stop recording before it gets too longer.
Yeah.
Sorry, we got overexcited. All right, bye everybody. I'll just stop recording.